Okay, in our next chapter, we're going to talk about the nutritional requirements for bacterial growth. Okay, we'll talk about the impact of nutrition on microbial growth and differentiation, which is change that the organism undergoes over time. We'll talk about the different physiological needs that support different types of bacteria and understand how metabolism and growth patterns can ID particular pathogens and talk about biofilms. In this section, we'll compare heterotrophy, autotrophy, phototrophy, and chemotrophy, talk about the nitrogen and carbon cycles, and talk about biofilms and their relevance to infectious disease. So, one of the things that we have to appreciate when we talk about any organism is that the organism is what it eats. Okay, so the building blocks that make up the structures of a particular organism are consumed in their diet. All right, essential nutrients are substances that bacterial cells need for their growth, get by essentially taking them in from the outside. Many bacteria require only elemental building blocks to produce all chemical compounds required for life, and those would be um, uh, bacteria, for instance, that can carry out um, chemotrophy or photosynthesis. Okay, because in photosynthesis you can take carbon dioxide and water and turn that into sugars, and then really everything else you need um, can be acquired by taking in those components from the environment in which you live. Generally, if you're talking about a bacteria, it's going to live in a liquid or a semi-solid environment. Okay, some bacteria live on actual solid material and acquire stuff from the air or from the rock that they're attached to. Okay, elements that are critical for life: carbon, nitrogen, oxygen, phosphorus, and hydrogen, and those are the building blocks that go into proteins and DNA and RNA and um, other structural components like carbohydrates and lipids. Okay, they also need sulfur, magnesium, iron, potassium, and then a variety of other trace elements, and they get these again by transporting them across their outer surface through either active or passive transport mechanisms. Some microbes require compounds known as growth factors that you have to add to the culture before they'll actually grow. Okay, examples, and again, we're talking here about the use of a growth factor in a lab setting, right? The growth factors that you need in the environment for the bacteria that are found out in the wild are naturally occurring in those environments. Okay, so example would be Shigella, right? And the growth factor is something called NAD, which is essentially a B vitamin. All right, Haemophilus influenza needs hemin and NAD. Staph aureus has a variety of requirements. Legionella needs cysteine. Okay, Bordetella pertussis needs glutamate, proline, and cysteine. Um, and Mycobacterium tuberculosis and Mycobacterium leprae need NAD, alanine. Um, Mycobacterium leprae is not culturable in a lab setting because it likes to populate areas in the human body that are made of nerve tissue. Okay, so that's a unique situation. So we would call that a fastidious strain, right? One that's difficult to grow. And then strep pyogenes, glutamate, and alanine. Okay, these are just a few examples, right? Culture media provides the nutrients that bacteria need in order to grow, and you can have either complex or chemically defined media. In chemically defined media, you know the exact amount of every component of that media, okay, um, to to the gram or to the milligram, okay. There are no components in the media that have unquantifiable ingredients, all right. Whereas in a complex medium, you'll have at least one component, maybe more, that have unquantifiable amounts of certain components, okay, because they are by their nature um, extracts of other things that aren't perfectly chemically quantified, okay. An example of a complex media would be something like LB, Luria Bertani broth, right, because you've got the tryptone in the extract, and the exact composition of those two components isn't known, okay. Whereas in a defined medium like M9, right, we know exactly what goes into M9 media down to the milligram, okay. Another example of defined media would be this sulfur oxidizer medium, right? You note that every component, we know the exact chemical composition. But wherever you see the use of an extract or something like tryptone or something like a broth, okay, or another undefined component, that's why we call it a complex media, okay? Nutritional needs of certain bacteria, such as Rickettsia, are unknown or very complex. They can't be grown in culture, and we call those fastidious microbes, okay? Difficult to grow outside of their normal environment. Carbon is critical for life. We talked about why, because carbon can do the one thing in the periodic table that none of the other elements can do. It can make rings and chains with itself, and so the building blocks of biological molecules use carbon as part of their backbone because you can make all kinds of important tools for life using carbon as a basis, right? In proteins, for instance, it's part of amino acids, okay? We also find it in carbohydrates and lipids and so on, okay? Um, Carbon compounds are used for food, which stores energy and is a source of cellular building material for biomass, right? So essentially we get down to why an organism needs to eat. It needs to eat because it needs to get energy from the chemical bonds and the nutrients that it takes in. It also needs to eat in order to have building blocks to assemble the components that make up the organism, okay? So it's basically eating what will eventually become part of its structure, okay? Autotrophs make their own carbon compounds starting with CO2. An example of an autotroph would be a photosynthetic organism, okay? Heterotrophs get carbon compounds from other organisms. Light or chemical compounds can be used as a source of energy by certain living things. Phototrophs use light energy for power in order to generate carbon compounds, which can then turn around and have the energy stripped from them in a process known as cellular respiration in order to generate ATP, while chemotrophs use potential energy stored in chemical compounds as an energy source. Lithotrophs, for instance, use inorganic chemical compounds, and organotrophs use organic chemical compounds. Okay, you would classify, for instance, a human being as an organotroph and a chemotroph, right? Because we consume organic chemical compounds for power. Okay, examples of components that lithotrophs might use for power include hydrogen gas, hydrogen sulfide, ammonium, nitrate, and ferrous iron. Okay, all of those represent reduced forms of either those compounds or those elements, and so they can be oxidized, and when they get oxidized, they're going to give off energy that'll be funneled into some other source. Okay. Again, when we're looking here at these different prefixes as descriptions for how the organism operates, we need to understand that a lot of the words you see that describe stuff in microbiology, or if you end up taking anatomy and physiology later on, come from the Greek and the Latin. Okay, so a good idea for this course and for A&P, if you end up taking it, is to get access to a website for medical terminology so that you can understand these word roots, prefixes, and suffixes, and that makes it easier for you to understand what you're seeing in the textbook. Okay? Autotroph is going to have carbon fixed and assembled into organic molecules, while a heterotroph is going to steal that from some other preformed organic molecule. A phototroph uses light for energy, such as a photosynthetic organism, while a chemotroph uses chemical electron donors, examples being things like lithotropes. Um, the electron source in a lithotrope is usually a reduced compound or element, okay? Whereas in an organotroph, it's organic molecules that end up donating the electrons, okay? Nitrogen is critical for survival of any organism because it's a component of DNA, RNA, nucleic acids, and proteins. So without nitrogen, there ain't going to be any growth or reproduction going on, right? So how do we get our nitrogen, right? If we're an organism, we can absorb it across the cell membrane, but it has to be in a water-soluble form, okay, for most organisms, meaning generally has to be combined with something like oxygen or hydrogen in order for it to be organically available, because nitrogen gas is really, it's a very stable compound. It makes up almost 80% of the air we breathe, but in that form, it can't be used to create things like amino acids or nucleic acids or nucleotides, okay? So it has to be fixed, and there are special bacteria that can do nitrogen fixing by transforming nitrogen gas into ammonium. It becomes available not only to the nitrogen fixing bacteria, but to the entire ecosystem, okay? The plants, the animals, and the other microbes. You can see here with the nitrogen cycle, nitrogen fixing bacteria converting nitrogen gas to ammonium, nitrifying bacteria converting ammonium to nitrite, and denitrifying bacteria take the nitrite and turn it back into nitrogen gas, okay? If you look in the periodic table at the element nitrogen, you'll see that with 5 electrons in its outer shell, it can triple bond to itself. So N2 has a triple bond between the two nitrogens, and so it's very tough to crack open, okay? And so these nitrogen fixing bacteria do an amazing job of making the nitrogen available. Now, another way that the nitrogen becomes available is through things like lightning, okay? The lightning can take nitrogen out of the air and convert it into water-soluble forms, okay? Examples of nitrifying bacteria, okay? Rhizobium cells clustered on a clover root tip are shown here. The rhizobia are on the surface of the root, and they will start to invade the root and begin a symbiotic relationship beneficial to both organisms. And you might wonder, well, how is that? Well, the rhizobium, okay, are going to do the nitrogen fixing, while the plant is going to provide a source of organic fuel, okay, because the plant can carry out photosynthesis, okay. Root nodules, also shown on the right. After the rhizobia invade the plant root, symbiosis between the plant and the microbe make up the nodules, okay. So this is another example of a symbiotic relationship. We saw an example previously when we talked about mitochondria chloroplasts inside cells, okay. This is an example of a symbiotic relationship between a bacterium and a multicellular eukaryotic organism, in this case, a vascular plant, okay. We'll talk about pure cultures and how you get them, and distinguish between synthetic complex, selective, and differential media. We kind of already talked about that a little bit. And talk about how bacterial growth is measured, and explain the advantages of each method in use. Okay? So, in order to study bacteria, generally, you have to be able to grow it in the lab, all right? And the way you do that is through the use of a growth medium that contains the necessary nutrients to provide the building blocks and the energy that allow that microbe to grow, okay? It'll also have to provide the proper pH and the proper salt concentration, the proper, we call it osmolarity, okay? And you have to grow it at the proper temperature, and that's going to be different for different types of bacteria, okay? Bacteria will grow in culture if the natural environment can be provided, right? Either as a liquid or a semi-solid. and then we have to have the right temperature and pH, as well as the right atmospheric pressure, right? Oxygen and other gases can serve as electron acceptors for those organisms that carry out um, uh, what we would call aerobic respiration. All right, not all bacteria do aerobic respiration, but a fair number do. Okay, an example of solid media is shown here. Okay, you take a source of bacteria. It could be from anything, right? It could be from a soil sample, or it could be from the surface of your skin. Okay, it doesn't really matter what it is. And then you can streak it onto this sterile growth media using either a metal or a sterile plastic loop. Okay, with the metal loop, the way that you do the streaking is that you heat it in the flame until it glows red, and then you let it cool a bit, usually by plunging it into the sterile agar, and then you dip the metal loop into the liquid media or the sample, right? And then you streak back and forth across the agar, okay? And then you reflame that loop to re-sterilize it, and then you dip it in the agar to cool it again, and then you go across the first set of streaks, okay, to make a second set of streaks, and then you flame and streak again, and flame and streak again, and eventually you get to the point where you can see individual colonies, which represent an area where a single bacterial cell grew enough to make a visible cellular mass, right? So every cell in this little spot here came from one bacterial cell originally, okay? So you want to get them nice and separated like that, all right? And the reason for that then is that you can then take those colonies, you can pick them with something like a sterile toothpick or other sterile implement, and then you can grow that in a liquid culture or on another plate, and then you'll have a pure culture of that particular organism, okay, whatever it happens to be. So the streak plate method lets you do separation of colonies into pure culture, okay? So let's take a look at how a streak plate is done.
Hello there. First video, we're going to show you some of the basic aseptic techniques that we use in microbiology. You'll get to practice these plenty in class, but we want to give you a little video that shows you the basic techniques. First thing I'm going to show you how to do is to use the inoculating loop, which is a very typical tool we use to move bacteria from either a culture onto a plate, from a culture tube to another culture tube, from a plate to a plate. Now there's a transfer of bacteria from one area to the other. Most important thing is when you're using the loop, it needs to be sterilized. There's a proper technique for sterilizing the loop, which I'll show you right now. With the Bunsen burner going, we're going to heat the loop starting up closer to the handle and slowly moving our way down to the end, so that if there was any liquid here, it would first evaporate and then burn off any of the bacteria versus aerosolize and potentially spread any of the bugs into the air. So put the loop in the flame, you'll see it will get red hot. Slowly move it down the flame, which will kill off anything that's on it, and eventually ending with the loop, which will get red hot, particularly the first time you use it during the day. You don't know where it's been. And once it is sterilized, you pull it out of the loop. Now we need to let it cool off. Things to not do when you cool it off is don't blow on it. That won't help matters. Don't stick it into a beaker of cool water, because the water won't be sterile. Basically, you have to let it cool off in the air, or we'll show you a technique later where you can touch it to a sterile area on a plate or on condensation in the plate if you're in a rush. Okay, so what I'm going to do is first I'm going to move some bacteria from this culture I have that I started yesterday of E. coli, and do what is known as a streak plate onto this plate. Before we get started though, there's some important record-keeping information we need to talk about. Namely, on every tube or on every plate you need to record 4 important pieces of information about what's going to end up on the plate. Before you put anything in, you need to label the name of the organism, the type of media, in this case Na, which stands for nutrient agar, initials of who did the work, the spreading, SDM, that's me, and the date at which the inoculation was made. Trying to keep it around the edge of the plate so that anything that's on the plate will be easier to see once they grow. Same kind of thing is done on a label. 4 corners, the bacterium, the date, the investigator or the student, whoever it happens to be, and the type of media, and if it's nutrient agar or nutrient broth, you see you have all those pieces of information. Important to make sure you get those all on there. So I'm going to move the bacteria that I have growing in overnight culture of E. coli. You can see in this culture tube, which has these lids on it that slide up and down. They don't screw on, which is going to be important because there's a technique which allows us to open and close these with minimal chance for contamination. And move it on to the plate, which most of you are familiar with, a Petri dish that has a lid on the top and has nutrient agar on the bottom. It's stored inverted, and then we'll talk about that as we show you how to do that. So if you're right-handed, having an inoculating loop in your hand like this, not sort of grabbing it like this. It'll be a lot easier if you get used to holding it sort of like a pencil, particularly so you have these two pinkies available to open and close tubes, as you'll see in a moment. So first thing I'm going to want to do is sterilize the loop. Again, put in the flame, slowly bringing it down, and I'm going to get hot. Now that's sterilized. It's cooling. Talk about how to open and close the lids. Since I'm right-handed, I'm going to use my pinky of the hand that has the inoculating loop literally to grab the lid. Thing you want to get used to. Every time you open it, open, flame, flame, close. By flaming it, I'll show you what I say. I'll grab the lid, if I pull it straight off and then just run it through the flame, I can do the work I need to do, flame, and close the lid. The flaming heats the air above the liquid, which causes it to expand and push out, which minimizes the chance for contamination. Never want to set the lids down. You always have them in your hand so that these have all been through the autoclave and everything started sterile, so you want to minimize potential contamination. So my loop is cooled now. What I'm going to do is flame open, go in. It'll pick up a little loop of liquid, which you probably can't see. Flame closed. Now I can set this down if I want. Since I'm going to be doing a streak plate, I have a little area on this divided into 3 sections. What I'm going to do is first flip the plate over and then spread starting at the top back and forth to get down here to the middle, and then I'll sterilize the loop, and you see drag into it a little bit and try to spread it out. We're basically trying to spread the liquid out and eventually get isolated colonies. Again, we don't want to take the lids off of these ever. We want to tip it, basically flip and tip. And we'll flip it, tip it, get in there, streak it, and then put it back down. So I'll flip it over, I'm going to tip it up, get in, let the loop run gently on the surface all the way to the middle here, close. Now I'm going to re-sterilize this, and once you've used it and you know there's nothing up on the shaft of it, you can sort of go in quicker, get the loop. I'm going to turn the plate now 90 degrees, and I'm going to dip once into where I had left off before and then streak into this quadrant, and then sterilize and do the same thing again. Again, just tipping it, go in here into where I had left off, go into here and spread it out. Re-sterilize the loop. And I'll turn the plate again 90 degrees, and one last time I'm going to try to dip into the area where I just streaked and drag it out and dilute it even further into the last quadrant. Tip, go in here, streak it out, done. When you're done, sterilize the loop. This could now be set down and flip the plate. Plates always get flipped, stored this way, minimizes the chance of anything falling on the surface and contaminating it. So there is a way to move liquid from the culture to the plate. The next thing we were going to show you how to do is to prepare what's known as a slant. Here's a slant tube which has nutrient agar that was autoclaved, sterilized, and allowed to cool at an angle so that it has this slanted surface, which provides a large surface area for the bacteria. And we use this when we do put them in deep storage or to keep long-term cultures in the fridge, and some media is prepared on slants, citrate agar being one that you'll learn about later. First thing I need to do is label this. I have a label prepared. Important thing with a slant, because what we're going to ultimately try to do is streak the bacteria on this surface. If I put the label up here, it becomes very difficult to see. So you'll learn as from experience that the best place to label it is somewhere down at the bottom, below which you're going to actually have the streak, so that it won't be in the way when you're trying to get in there. The only difference between this tube, another difference between this tube and the culture tube we had, is that it has a screw top on it, so it's a little different in opening it. The technique will be basically the same, and I'll have an inoculating loop in my right hand, because I'm right-handed, and I'm going to use my pinky or my ring finger to open the tubes. Difference here is that instead of pulling the tube off, what I'll do is grab the top. Now you could try to unscrew it like a soda bottle or something, but it'd be too easy to drop the top. and then it becomes no longer sterile. So the trick that will sort of encourage you to get used to is hold the top, turn the tube, and you will be able to get to a point where you'll know that the top is off and is ready to, that you can easily pull the top off, flame open, do the work you need to do, put the top back on after you've flamed it closed, screw the top back in as opposed to trying to screw the top. Okay? So what I will do is move the bacteria from this nutrient broth into the slant tube, hoping to make sure to flame open and flame close each time. First thing I'll do is sterilize the needle, the loop. And again, I have a culture tube. As you get better with this, you'll be able to hold both tubes in your hand at the same time until you get comfortable with that. You can do it one at a time. So I'm going to flame open, do my work, flame closed. A loop full of liquid, flame closed. I can set this down now. I will, again, I'm going to unscrew it. I can tell it's ready to go. Flame open, literally loop in here and drag it across the surface carefully, pull it out, flame closed. And the last thing I need to do is remember to sterilize my inoculating loop so that I'm not going to contaminate the desk. All set.
Okay, some other types of media that are available out there include selective and differential media. Okay, in selective media, what we do is create an environment in which only certain organisms from a mixture of organisms are going to be able to grow because of the composition of the media, okay, and the conditions under which we grow them, right? The temperature and the pressure and all that, okay. In a differential medium, what will happen is that you can grow more than one type of organism, but the appearance of that organism will be different depending on the types of chemical reactions that it can carry out. So with differential media, you don't necessarily get a pure culture growing up like you can with selective media, but you can identify colonies that are made up of a single organism based on their appearance. For instance, their color, or maybe the color of the media surrounding them, okay? Oftentimes, for instance, one of the things that's used to differentiate different organisms in differential media is a dye that changes color depending on the pH, okay? And some organisms in that media might create acids that'll drop the pH and have it, that colony turn a particular color, and then other bacteria won't, okay? An example, right? MacConkey medium is both selective and differential. Okay, this is MacConkey agar here. It's selective for the growth of gram-negative bacteria because it contains bile salts and crystal violet, and that prevents the growth of gram-positive bacteria because it interferes with the ability of those bacteria to create their cell wall. Okay, lactose fermenters such as E. coli degrade lactose and secrete acids in the media that lower the pH around the colony, and the acidic pH lets the pH indicator neutral red enter the cells and produce a red colony. All right. Non-fermenters such as Salmonella enterica don't ferment the lactose. No acids are produced, and so the neutral red does not enter the cells, and the colonies end up being white. Okay, and you can see some examples of white colonies here, right, and then fermenters here in the dark red. Okay, so if you were to pick from this dark red colony, all right, you would grow up a pure culture of lactose fermenting bacteria of some type. Okay, and if you were to pick from this white colony, you would grow up non-fermenting bacteria of some particular type or strain. Okay. One of the things that we like to do when we work with bacteria is to determine the number of cells that are in a liquid culture sample at a particular point during its growth. Okay, basically, if you consider a liquid medium, all right, that has a certain amount of nutrients in it, and you were to inoculate that liquid medium with a certain number of bacterial cells, all right, you could take that liquid media and then through proper conditions, such as, let's say, shaking at 37 degrees, cause growth of that inoculum to begin, right? And so what would happen initially is a few cells will become a few more cells, and then if we were to determine the number of living cells in the media, what we would see basically is a graph. We can do this in let's do blue. Okay, you see a graph. Okay, there's a vertical axis. There's a horizontal axis. This is time, all right, and this would be number of cells. Okay, and so what you would see in a liquid growth medium is you would see a period called the lag phase, where we're starting to pick up cell numbers, and then we would hit what we call the logarithmic phase, and then we would have a plateau phase, okay, and then we would have a death phase on the other end, all right, and eventually all the bacteria would be dead because we would run out of nutrients, okay. And so usually when you're working with a bacterial culture, if you want to work with a healthy cell population, you like to take cells from that culture at a time when the culture is happiest, and that usually is sometime around what we would call mid-log phase. Okay, so you would determine the amount of time that would need to pass. You would figure out when that mid-log phase was, right, and then that would tell you what your cell numbers would be in that in a particular volume of sample that you would take. Okay, the reason you don't want to take it at what we would call plateau or in the death phase is because a lot of your cells are going to be sick, or they may be undergoing a process called sporulation, where they're kind of going into hibernation and they're making bacterial spores. Okay, one of the ways that we can count cells is through a direct or an indirect cell count. Okay, in a direct cell count, what we do is we take a certain amount of the liquid media, and then we put it on something called a hemocytometer, which is a sort of a microscope slide that has a grid on it, and then we know the area of each of those little grids, and then we count the number of cells in a particular area of the grid, and that allows us to compute the number of cells per unit volume in that particular liquid media. Okay, that's called a direct cell count. Now, one of the things we don't get from a direct cell count is how many of those cells are alive. Okay, we just get cells. Some of those cells could be dead, right? In an indirect cell count, we do something called FACS, or we use a spectrophotometer. FACS stands for fluorescence activated cell sorting. Okay, and what it does is it is able to count particles that have that have the ability to deflect a beam that's in a suspension. Okay, and give us based on um, the, the particles that are in the sample stream, a count of the number of particles that passes through. Now again, we don't know, well, we can know in some cases, but for the method we're using here, we don't necessarily know whether or not the cells are alive or dead. Okay, there are some methods that do allow us to know that, but they're more complex than what we're looking at here. But anyway, you pass this flow through, the laser hits it, okay, and then you end up with a certain amount of this going into waste, right, and a certain amount of this going into the cell collector so you can recover your sample, okay, and the reason for the deflection is because of the voltage that goes across this little chamber, all right? Okay, so there's a certain charge associated with the particles that contain cells, okay? Turbidity is different. Turbidity is just the ability to scatter light that's passed through the sample, okay? So what you do is you take a certain amount of liquid sample, and what you basically are measuring is how cloudy it is, because the less light that passes through the sample from one side of the sample, usually contained in something called a cuvette, which looks like a little glass or plastic box, okay, to the other side of the cuvette, the amount of light that passes through is going to be less the more turbid it is. So the cloudier it is, the less light's going to go through, and you can use that as an estimate of the number of cells in the medium, okay? let's take a look at um, dilution streaking. Okay.
Agar medium in a petri dish provides a large flat surface on which a mixture of microorganisms can be streaked to separate individual cells. In the dilution streaking technique, an inoculating loop is sterilized in a flame and allowed to cool. The loop is used to collect a drop of liquid from a bacterial culture. With the loop held flat on the agar, the drop of liquid is streaked across a region of the agar plate's surface. As the loop moves, organisms begin to rub off the loop. The loop is sterilized and cooled again. This time, the loop streaks over the first pass, collecting a small number of bacteria and spreading them into a fresh area of the dish. The procedure is repeated again. Toward the end of the streak, few bacteria remain on the loop, so at that point, individual cells will land and stick to different places on the agar surface. If the medium contains the proper nutrients and growth factors, a single cell will multiply into many millions of offspring, forming a colony. In general, a pure culture can now be obtained by touching a single colony with a sterile loop and inserting that loop into fresh liquid medium. It is important to note that the one cell equals one colony paradigm does not hold for all bacteria. Organisms such as streptococci or staphylococci usually occur as chains or clusters of several cells. A cluster of 10 or so cells, called a colony-forming unit, or CFU, may give rise to a single colony.
So basically the idea behind this method is that you take a sample of your culture, all right, at a certain time, and then you dilute that sample, usually a series of 10-fold or 100-fold dilutions, and then you plate those dilutions onto nutrient media, like nutrient agar plates, and then you count the number of colonies that grow up, and that gives you an estimate of the number of living bacteria in that sample. Okay, so if you were to take, for instance, let's say that you took 20 microliters of a log culture of LB, of say organism like E. coli, all right, and then you were to dilute that 1,000-fold, okay, and let's say you got 5 colonies growing up on that 1,000-fold plate. You used 20 microliters of the 1,000-fold dilution, okay? Then what you could do is take the dilution factor and work backwards and determine how many bacteria there were in the original 20 microliters that you took out of the culture, okay? So let's say that you inoculated 20 microliters into, you did a 1 to 10, all right? So you put 20 microliters into 190 microliters of you could say water, okay, or you might use media, okay, and then you were to repeat that 2 more times, okay, and then you would have using a fresh transfer pipette for each one, okay, and mixing in between, and then you would end up in your third dilution. The first dilution would be 1 to 10, the second would be 1 to 100, the third would be 1 to 1,000, right? So if you took 20 microliters out of that 1 to 1,000, that represents 10% of that whole volume that's in that dilution, right? So if you were to grow 5 colonies from plating 20 microliters, which is 10%, that means in that entire dilution tube, okay, you would have, if 10% is 5, you're going to end up with 10 times more than that, which would be 50 colonies in that dilution. Well, since that's a 1 to 1,000 dilution, you got to multiply the 50 by 1,000, so you get 50,000 living cells in that original 20 microliters. Okay, and that would, and you plot that depending on what time points that you took the liquid culture at. Okay, so that's a way to determine the proper method for growing the bacteria that you're studying. Okay. Okay, we'll talk about population growth and phases of a growth curve. We kind of already looked at that a little bit. And we'll describe generation time for a bacterial population. And we'll talk about how bacterial growth correlates to disease and the purpose of continuous culture and how it correlates, for instance, to the human digestive tract. So bacterial growth is measured at the population level, and the growth rate is a measure of the number of cells in a population over time. Most bacteria reproduce by binary fission, which we've already seen details of, okay? And binary fission can either be symmetrical or asymmetrical, all right? Symmetrical, the results of the binary fission are exactly the same size. Asymmetrical, you end up with different products at the end of that fission, okay? Eukaryotic microbes always divide by mitosis, okay? So you can see here the different phases, right? Lag phase, log phase, stationary, and then death phase, right? Kind of like I drew earlier. And I told you that generally speaking, all right, you want to take cells from mid-log, all right, in order to be working with a healthy population, okay? And that's because the rate of growth is greater than the rate of death at that point, which means most of the cells you're going to be using are going to be alive and happy, okay? At stationary phase, the growth rate and the death rate are the same, okay? That's why the turbidity, that's why the number of living cells isn't increasing, but it also isn't going down. And in the death phase, the rate of death is greater than the rate of growth, okay? Phases of bacterial growth in a typical batch culture shown here. The shadings under the curve indicate where those different phases are. Note in this graph that the y-axis is a log scale and reflects an order of magnitude difference between cell numbers instead of a simple increase in cell number. One order of magnitude difference equals a tenfold increase in cell number here. And the reason you do that is because we didn't plot the number of microbes on a log scale on the vertical axis. The graph would be incredibly long, okay? And so what we do is we, the differences between the divisions here are tenfold divisions, okay? So 10 to the 7th is tenfold higher than 10 to the 6th. 10 to the 8th is tenfold higher than 10 to the 7th, and so on, okay? With unlimited resources and the right environmental conditions, bacteria can generate a new generation of cells at a constant time interval known as the generation time, also called the doubling time, okay? If the generation time is known, it's possible to estimate the number of cells in the population using the expression n sub 0 times 2 to the n, where n sub 0 is the number of cells the culture started with, okay? So that would be your inoculum, okay? And the little n refers to the number of generations that culture has undergone, okay? So E. coli has a generation time of 20 minutes. How many cells will there be in a culture that begins with a single cell and grows for 4 hours, all right? So n sub 0 would be 1, okay? 2 to the n, what would that be? Well, how many 20-minute periods are in 4 hours? Well, an hour is 60 minutes, right? So there's 3 20-minute periods in an hour, right? So you take the 3 and multiply it by 4. That's 12 20-minute periods. So it'd be 2 to the 12th. So it'd be 1 times 2 to the 12th, okay? And if you do that, that equals 8,192, okay? The continuous culture conditions that you get in something called a chemostat are designed to try and keep the bacteria in log phase in perpetuity, all right? And the way that works is that you dribble in fresh media as spent media dribbles out, okay? And that way you always have a culture that has a population in optimal health, okay? The human GI tract is engineered like a chemostat in that new nutrients are always arriving from the throat and equal amounts of bacterial culture exit in fecal waste, right? So that's an example in the human body of something that's a lot like a chemostat, okay? They also use chemostat type conditions with other types of microbes like yeast, okay? Basically, making beer is a little bit like a chemostat, okay? Um, you want the population to not start to go into death phase, okay? Or even to get to plateau. We'll talk about how environmental changes change the shape of the growth curve, and we'll list different classes of microbes based on what they like to live in and like to take in. Okay, that's called their niche. And we'll ID biological properties that allow different classes of microbes to grow in extreme environments. Okay, so bacteria are very diverse. They can live in almost any environmental condition that you find on planet Earth. You can find them near hydrothermal vents, which have basically the same composition as boiling battery acid. You can find them deep in the Arctic ice cores. Okay, um, it's amazing where you can find bacteria that you wouldn't think there could be possibly anything that could live. Okay. In addition to food, bacterial growth requires the proper temperature and pressure, the proper osmotic balance, and the appropriate acidity, the appropriate pH level. Okay? The optimal conditions will vary depending on the kind of organism we're talking about. Okay? A psychrophile, with regard to temperature, tends to optimize at temperatures around 15 centigrade, right? A mesophile optimizes close to 40, okay? A thermophile digs around 65, right? And then a hyperthermophile is going to like 90. Now, why such a vast difference in temperatures for these different organisms? Well, they use different tools for carrying out their life processes, right? The psychrophile, for instance, uses protein tools for life that dig cold temperatures, that work best at cold temperatures, while the hyperthermophile produces tools for life that are best at very, very high temperatures, okay? And the reason for the structural differences, the reason that these structural differences work at these different temperatures is due to the amino acid composition of the proteins. For instance, a lot of the proteins in hyperthermophiles have a lot of disulfide bonds in them so that they don't come apart at high temperatures, whereas in the psychrophile you have fewer of those. Okay? The same is true for any other parameter. We could do an optimum for the pH, we could do an optimum for the temperature like you've seen here, and we could do an optimum for the osmolarity of the media. Okay? And all of these organisms would have conditions that are the best at which they grow. Okay? So each curve represents the temperature growth range for different bacterial classes. The growth increases linearly with temperature until you get an optimum, and then it decreases. Why does it decrease after the optimum? The bacteria proteins begin to denature, right? And so the chemical processes they carry out slow or stop, and the bacteria dies. Okay? These curves are approximations, right? Real ranges within a class will vary, and that's because the tools of life within a class will vary because the genetic information to build those tools will be a little different from organism to organism. Okay? Thermophiles and extreme hypothermophiles, such as archaea, like extreme conditions, like very, very high or very, very low temperatures, or very, very high or very low pHs. Okay? Psychrophiles like ice, whereas hypothermophiles like those hydrothermal vents, which are basically like the openings of a volcano deep under the sea. Okay, so they're belching hydrogen sulfide, and the pHs are very low, and the conditions are quite hostile. Okay, an example shown here, right? Yellowstone National Park hot spring and an iceberg where psychrophiles like to live. Okay, bacteria from the South Polar snow on the surface of a membrane filter are shown here, right? And these organisms have yet to be fully classified. Pressure is another one, right? Barophiles like very, very high pressures. The deepest part of the ocean is at the bottom of the Marianas Trench, which is a depression in the floor of the Pacific east of the Mariana Islands. The Mariana Trench is about 1,500 miles long and about 44 miles wide. Near its southwestern edge, it's about 210 miles southwest of Guam, and you have here the deepest point on Earth, okay, referred to as Challenger Deep, and it's about 7 miles under the surface of the ocean, okay, where the pressures are 1,000 times higher than what we experience on the surface of the Earth at sea level, okay? So that's some extreme sets of conditions, right? Most organisms at sea level would be crushed by those extreme pressures, but the organisms that live in the trench have adaptations that allow them to survive those extreme conditions, okay? Another one is osmotic pressure, okay? Halophiles, for instance, can survive with 30% salt concentration. Most halophiles are archaea, right? That special branch of prokaryotes that is a little bit more similar to eukaryotes than it is to bacteria, okay? The halophilic salt flats shown here in Nevada are pink because of the high numbers of halophilic bacteria that grow here. Halobacteria species shown in the little inset have a width of about 0.8 microns. Okay, so again, adaptations that allow it to hold on to its fluid volume even though the environment outside is extremely high osmolarity, meaning very little water and a whole lot of solutes. Okay, pH is another one, right? Note that pH and pOH are reciprocal values. We talked earlier about the fact that the law that always holds in water is that pH plus pOH equals 14. And we talked about the fact that you can go from pH to the molarity of protons by just understanding that all you have to do is take the pH and turn that into the negative exponent that you raise 10 to. Okay? So what's happening here is that the concentration of protons is increasing as we go up, and the pH goes down, and it's decreasing as we go down, okay, and the pH goes up, all right? And that's because pH is the opposite of the log to the base 10 of the molarity of the protons, okay? We'll talk about anaerobes and aerobes and describe their culture conditions. And we'll talk about how aerobes and anaerobes can produce disease and talk about the differences between respiration and fermentation. Okay? So aerobic just means needs oxygen or uses oxygen. Okay? A strict aerobe absolutely has to have oxygen to survive. It successfully detoxifies reactive oxygen species, which can form free radicals and damage healthy molecules inside the cell. It has to have oxygen to live as a result, and it includes all groups of life other than bacteria. Okay? Strict anaerobes don't require oxygen for energy metabolism and are unable to detoxify reactive oxygen species, so oxygen is toxic to them. They survive only in environments that don't have any oxygen. Only bacteria populate this category as strict anaerobes, all right? There have been a few organisms that have been found that are anaerobic other than bacteria, but we don't know if they're strict anaerobes yet, okay? These are relatively recent discoveries, okay? What you're looking at here is a liquid culture, right? As oxygen diffuses and equilibrates with air, oxygen levels in different areas of a simple nutrient broth will decline the further down the tube you look. If we inoculate a series of tubes like this with bacteria having different oxygen requirements, it'll result in the anaerobes growing at the bottom of the tube, the strict anaerobes. The microaerophilics will grow in the middle, and the strict aerobes will grow only at the top. Facultative organisms will be everywhere. Okay, so what do each of these terms mean, right? Aerobic has to have oxygen to live. So why is it near the surface? Because the oxygen's dissolving in the liquid at the surface um, at about 9% if it's water, okay? And that's enough for these to grow, right? But they can't grow at lower oxygen concentrations, so you won't find them in here or down here, all right? Microaerophilics need low oxygen concentrations to grow, okay? So they'll be found in this middle zone, and strict anaerobes are going to be the furthest away from the oxygen at the very bottom of the tube. Facultative, all right, just means that these guys are switch hitters, okay? They can go totally without oxygen, they can go with low oxygen, or they can go with high oxygen, okay? So they can do it all, okay? So when we say aerobic, one of the terms that we use for strict aerobes is obligate aerobes. And when we say strict anaerobes, we say obligate anaerobes, okay? Obligate means that you have to live under those conditions, right? Whereas facultative means you can switch off, okay? And you can see some of the different oxygen concentrations here in the growth media, okay? Microaerophiles are aerobic but reactive oxygen species. If they're build up too high of a concentration, can be toxic and kill them. And they survive in environments with low oxygen. Aerotolerant anaerobes are anaerobic but not as susceptible to these oxygen species. They don't have an enzyme called catalase, which is one of the ways that we get rid of these free radicals that come from oxygen. And they prefer anaerobic conditions but can survive low oxygen levels. Okay? Well, facultative anaerobes can do anything, right? They can either take or leave oxygen. They can switch off. It doesn't make any difference. Okay? What you can see here are some different methods of culturing different organisms, right? You've got an anaerobe jar for the growth of strict or obligate anaerobes, okay? And an anaerobic chamber, right? You can see here that it's equipped with ports that remove the atmosphere through a vacuum and replace it with nitrogen and carbon dioxide gas, right? Sort of a low-tech way to do this, less fancy way to do this, is to make what's called a candle jar, okay? And then what you do in a candle jar is you have sort of it looks like a tall glass jar, okay, or a metal jar, and it has a metal or a glass lid. And then what you do is you stack your plates with your anaerobic cultures in there, and then you put a candle on top of the top plate, and you light it, and then you put the lid on, and what the candle will do is as it burns, it'll burn up all the oxygen inside the jar, and you'll be left with an anaerobic environment in which those bacteria can grow, okay? So the anaerobe jar shown here lets plates be inoculated. Once in the jar, we add water to the gas pack, and the top is sealed shut. Hydrogen and carbon dioxide come from the foil packet, and the hydrogen reacts with the oxygen in the chamber and makes water and removes free oxygen from the chamber, okay? An anaerobic chamber with glove ports is shown below, right? And this is a more expensive way of growing the culture, okay? So you can have systems that could cost anywhere from thousands of dollars to maybe just under 100 in the case of something like a candle jar, okay? And they're all different in terms of their effectiveness, okay? You can see some examples of aerobic and anaerobic microbes here, okay? This is 6.4 in the text. And I would just, note the characteristics and example organisms in each, okay? And understand why that affects where they're found out in the environment, okay? Okay, next we're going to talk about biofilms and explain the importance of biofilms in infection, talk about sporulation and how spores impact infection, okay? So biofilm is basically a bacterial community, okay? And it's thought to be the precursor to tissues, okay? In a biofilm, what happens is you have organisms that specialize in different tasks that help the community survive, right? Some are good at building a home, some are good at defending against attack, and some are good at acquiring and distributing nutrients, okay? Biofilms have networks of communication between them, and they have a variety of environments within the biofilm that create different niches for different types of bacteria by changing, for instance, the oxygen concentration, the pH, and so on. Okay. What makes biofilms so tough to deal with from a disease aspect is because they're not a pure culture. Okay. What may kill one part of a biofilm doesn't necessarily wipe out the rest of the biofilm, and the biofilm can adjust to any chemical used to try and attack it. For instance, like an antibiotic. Okay. So let's take a look at the formation of a biofilm.
Bacteria were once thought of as unicellular organisms incapable of differentiation or communication. But we now know that many, if not most, bacteria form specialized surface-attached communities called biofilms. Indeed, within aquatic environments, bacteria are found mainly associated with surfaces. This biofilm is a plaque that forms on the surface of a tooth. Plaque is a mixed species biofilm, but single species biofilms also form in nature. For example, single species biofilms of Pseudomonas aeruginosa can form on the lungs of cystic fibrosis patients. The first stage of biofilm formation occurs when a specific environmental signal triggers unattached planktonic cells to attach to nearby inanimate surfaces by means of flagella, pili, lipopolysaccharides, or other cell surface appendages. The attached cells begin to coat that surface with an organic monolayer of polysaccharides or glycoproteins, to which more planktonic cells can attach. As the cells enter into biofilm formation, they no longer maintain their flagella, and instead may move along surfaces using a twitching motility that involves the extension and retraction of a specific type of pilus. Ultimately, they stop moving and firmly attach to the surface. As more and more cells bind to the surface and divide, they form microcolonies and can begin to communicate with each other by sending and receiving chemical signals in a process called quorum sensing. These chemical signals are continually made and secreted by individual cells. Once a population reaches a certain number, analogous to an organizational quorum, the chemical signal reaches a specific concentration that the cells can sense. This concentration triggers genetically regulated changes that cause cells to bind tenaciously to the substrate and to each other. Quorum sensing may influence the amount of extracellular matrix produced by the cells. This thick, slimy material consists of polysaccharide polymers called exopolysaccharides and entrapped organic and inorganic materials. These exopolysaccharides increase the antibiotic resistance of residents within the biofilm by limiting antibiotic access to the biofilm center. The chemical signal molecules also stimulate production of some antibiotic resistance mechanisms. As the biofilm matures, the amalgam of adherent bacteria and matrix forms columns and streamers, creating channels through which nutrients flow. Although most of the bacteria adhere to the biofilm, some planktonic cells leave for new environments. Here is an image of the lumpy, uneven biofilms created by a mucoid environmental strain of P. aeruginosa. Biofilms allow bacteria to stay where food is plentiful. Why should a microbe travel off to hunt for food when it's already available? Once nutrients become scarce, however, individuals will detach from the community to forage for new sources of nutrients. P. aeruginosa produces an enzyme that can strip away the exopolysaccharides and will resynthesize flagella. Cells leaving the biofilm revert to their flagellated planktonic forms and can swim to new locations where nutrients are more plentiful. A hallmark of infectious diseases in which biofilms play a prominent role is their chronic nature. A biofilm infection may linger for months, years, or even a lifetime.
Okay, endospores are an adaptation to low nutrient conditions in the environment. Okay, some bacteria have the ability to form these endospores, which are the bacterial equivalent of a seed for a plant. Okay, and they're able to survive extreme conditions. And then once the other components of the environment that promote growth come back, the endospore can germinate and vegetative growth can begin again, and you'll have a rootin' tootin' bacterial cell where before you just basically had this little spore. Okay, so endospores are very tough to kill, and that's one of the reasons why we have to use techniques like autoclaving in order to get rid of endospores on materials that can stand it. And basically, autoclaving uses high heat and pressure and steam in order to destroy endospores, right? Whereas vegetative cells are killed with much milder conditions. Okay, bacteria in endospore form are dormant and don't need nutrition or energy. Examples include Bacillus anthracis, Clostridium botulinum, and Clostridium tetani. Okay, as you can probably guess, Bacillus anthracis is the agent of anthrax. Clostridium botulinum is the agent of botulism, and Clostridium tetani is the agent of tetanus. Okay, and these are the results of toxins that they make. Okay, the endospore state can last almost indefinitely. Okay, before reactivation occurs. Let's take a look at endospore formation.
When starved for nutrients, certain gram-positive genera, including some important pathogens, can develop dormant heat and desiccation-resistant spores. Most of our knowledge of bacterial sporulation comes from the gram-positive soil bacterium Bacillus subtilis. When growing in rich media, this microbe undergoes normal vegetative growth. B. subtilis can replicate every 30 to 60 minutes. In contrast, starvation initiates sporulation, an elaborate 8-hour genetic program that ultimately yields a spore. In the first stage of sporulation, the septum forms near one pole of the cell rather than in the middle. The DNA replicates and extends into an axial filament that spans the cell's length. During the next stage, the septum separates the forespore, which will ultimately become the spore, from the larger mother cell. DNA is pumped through the septum until each compartment gets a chromosome. The mother cell now engulfs the forespore, surrounding it with a second membrane. The mother cell chromosome is eventually destroyed. A cortex, made up of a thick peptidoglycan layer, is placed between the two membranes surrounding the forespore. This cortex will be important in providing heat resistance and maintaining dormancy to the spore. Layers of coat proteins are deposited on the outer membrane. In the next stage, dipicolinic acid and calcium are incorporated into the spore. Dipicolinic acid is important in maintaining dormancy and in promoting spore dehydration, a characteristic that makes a spore heat resistant. The mother cell, now called a sporangium, dies and releases the mature spore. Spores are resistant to many environmental stresses that would kill vegetative cells. The nature of this resistance is due in part to the spores having only 10 to 30% of a vegetative cell's water content. But spores also contain small, acid-soluble proteins that bind to and protect DNA from damage by ultraviolet light and various toxic chemicals. Fully mature spores can exist in soil for at least 50 to 100 years and have been known to last thousands and even millions of years. Once proper nutrient conditions arise, another genetic program, called germination, is triggered to wake the dormant cell, dissolve the spore coat, and release a viable vegetative cell.
So what you're seeing here is just an example of the different growth stages of a bacteria, depending on whether it's going to go into sporulation or whether it's going to do vegetative growth, right? In vegetative growth, we're just going to undergo binary fission and make two brand new bacterial cells that have the same genetic composition as the original. So that's the prokaryotic version of mitosis, okay? But when we're doing sporulation, right, what happens is that nutrient conditions, water availability, or other environmental factors are not optimal, okay? And so we have to go into basically dormancy, right? So the first thing that happens is you get this septum that begins to form. It separates into a forespore and a mother cell. The DNA is pumped through the septum into each compartment, getting a chromosome, right? So there's going to be DNA that's going to be in the vegetative cell and some that's going to be in the spore, all right? And that the information content is going to be the same. And then in the next stage, the mother cell eats the forespore, surrounding it with a second membrane, okay? The chromosome of the mother cell disintegrates, all right? And then what you end up with is the genetic material, all right? You've got cortex around the genetic material, all right? And then the forespore that has the peptidoglycan layer ends up also with a temporary membrane around it, okay? This gives time for a spore coat to form, okay? Coat proteins make up the spore coat. And then the membrane is going to disintegrate, okay? And then the spore will last as long as it needs to last, okay? Diplocholic acid and calcium end up in the core of the spore, and that's sort of a preservative for the stuff inside it, okay? And this little guy can survive extreme conditions. The vacuum of space, extreme cold, extreme heat, up to a certain point, okay? And then when the conditions are right again, we can restart vegetative growth, right? Okay. So this brings us to the end of microbial differentiation. You've seen the illustrations here in the presentation. You can go back and look at them again. I'll put the links in the PDF, and I will see everybody in the next podcast. I thank you for listening.